for organizing this. Can you hear me, everybody? And you can see me.
Yep, good job.
Okay, great. Great. Over the today and tomorrow, two other people from DataJoint will join the presentation. So Kabi and David will present. So I will give a quick overview over the next 9.5 minutes, and we'll go from there. So this is a neuroscience experiment from a few years ago, and just to convey the feats of data engineering that students and postdocs who are not primarily software engineers and data systems engineers have to accomplish to collect the data to organize experiments, combining sometimes a dozen of different modalities in a single study: electrophysiology, whole cell electrophysiology, imaging, behavior, recording behavior, also stimulation, optogenetics, all in a single study and in various ways, in various permutations. Maybe each experiment doesn't have all of them, but the overall conclusion depends. The overall findings depend, and so on using all the modalities. So this continues to increase. So another experiment we participated one was IR microns. The data were just released 2 weeks ago on July 29th, and so you can go to micronsexplorer.org or micronsdatajoint.io to interactively play with a single mouse's data. This is called the platinum mouse. It's probably, and please correct me if this claim is wrong, but it's probably the most studied tissue, 1 cubic millimeter of tissue in human history. So we at Baylor, the Baylor team imaged with calcium imaging a full cubic millimeter with various visual stimuli, and characterized every cell, about 100,000 cells. And then that cubic millimeter was imaged with 40 nanometer resolution, and then the data were segmented at Princeton with identifying all the cells, all the segments of all the cells. So the data available, a lot of it was organized with DataJoint, and so in micronsdatajoint.io you can interactively work with these data. So and since then, the progress doesn't stop, and I think it will continue to increase. Just looking at the amazing uptake of neuropixels by the neuroscience community, with now hundreds of labs using it, and now with the shift to NeuroPixels 2.0, the amount of data and the complexity of data will only continue to increase. And there is a shift toward much faster types of experiments where automation is very critical. This is in the industry, there is this concept of a digital twin, when a digital representation of a complex system, such as a jet engine, for example, is built in order to be able to keep track of everything that's happened to it, and also run simulations to see what effects it can have. And so the same thing happens in neuroscience, like this one paper, for example, and many similar papers are now coming out where the digital representation of the studied circuit is built in artificial neural networks. And then in a tight loop, you can run in silico experiments and match them with neuroscience experiments. And so this creates the need. So on the same mouse, the data needs to be processed the same day. So the next day, the experiment can be repeated on the same animal with new parameters driven by the simulation. So what's the fundamental problem we're trying to solve? So this is very difficult from the engineering perspective to organize it, and there's a lot of wasted effort with people reinventing how to organize all the processes of data collection and analysis just to conduct a single study. And then sharing data, one problem with sharing data is that it's an extra effort. Sometimes it takes weeks or months to package the data collected from a study. So our solution has been DataJoint, which is basically a framework for formalizing and representing one of the biggest assets in science. It's the scientific workflow. The overall process, all the steps. At one point, we tried to estimate the number of steps that you have to perform to conduct an experiment, and it's in the order of like 400 steps. And if you include preparing pipettes and you include the surgery and everything, if you had to write out what you did for your experiment, it will be hundreds of steps. And that's actually an intellectual property. That's like a huge asset that there's not a very good way of sharing it. It's captured maybe in the methods section, very superficially. So DataJoint is a framework for capturing the manual steps, the data management, and the automated analysis in a single framework. And so the framework itself is on datajoint.org. It's been in a long time in development, and we're committed to keeping it as an open standard. The word standard has been used a few times, but open standard in the technical term that there's community input to drive its development. And then we have the NIH-funded U24 project to produce specific solutions for each neurophysiology experiment, each type of neurophysiology experiment, and we'll talk more about that. And as part of sustainability, we are a company. We provide managed services to run the whole thing. So all the resources can be run by each lab separately, or we can help, or we can also manage the entire workflow. So a little bit of history. So it started as a personal project for my own experiments in 2009, and then the lab adopted it. It was an Andreas Tolia's lab at Baylor College of Medicine. By about 2011, the lab adopted it, and then other labs started joining. And in 2016, we got funding from DARPA to start a company. We called it Vase LLC. We now go by DataJoint, since most people know us by our product. And then a lot of, as we started, multi-lab projects started using it. This is just a flashback from documentation from 2011 of the basic concepts, basically representing workflows for calcium imaging. So there's a lot more than FAIR in organizing that part, the active part of the project. FAIR is designed for sharing data once the data has been packaged for outside, for mostly for sharing with friends, with collaborators. To organize the workflow with computations and everything inside as you run the project requires structured data integrity, data consistency, meaning that many agents interacting with the data, reading and writing, should not the entire data set needs to appear as a single consistent copy. It needs to manage the workflow and provide efficient queries with indexing of the data and natively support distributed computing. And so this is described now in this preprint as part of the U24. And so even organizing a small study, it's amazing how much expertise you need to put together in order to organize it well. You need the IT infrastructure, data engineering operations, data science tools, and then actually run the experiment workflow. So DataJoint itself is a framework for formally expressing all the data engineering operations. As your project grows, you will find yourself doing more and more of data engineering, just moving files around, transferring them, transforming them from one format to another. The more you do this, the bigger the team, the more distributed the team, sending the data back and forth. Some people report, and I don't know if I've seen that, but they report 80% of the time spent is more on the data engineering. And so what DataJoint is, it's basically a relational database language that extends the relational concept for scientific computing, to represent automated workflows. So each table represents either a manual data collection step or an automated analysis step. And it allows storing complex data such as calcium traces, movies directly in the database or block storage. So a typical architecture for a medium size, you can start very simple with a laptop on a single laptop. So this is something I would like to highlight. Many big projects use DataJoint, but I use it for very small projects as well, and you can just get started on your laptop. But where it excels is in big projects. This is a medium-sized project. The architecture consists of a database, some servers for ingesting the data, metadata interface, which is usually a web-based GUI that interacts with the database, and client APIs for data analysis and navigation. So we support many different projects, but these are the big multi-lab consortia with whom we have active projects right now. So they rely on DataJoint to organize these multi-lab collaborations. So DataJoint Elements is the funded project. So until now, or until recently, a lot of labs would start from scratch. Most of these projects are open source, but they still started from scratch because they all feel like their experiments are different, even though they're doing calcium imaging and electrophysiology. So we decided to start, or we proposed to provide good starting points. And so we are now in year 2. We just started year 2. In year 1, we completed array electrophysiology and calcium imaging. And so there's a training background. We also created a new graphical user interface, DataJoint Lab Book, that allows interacting. There's a backend called Ferris. Lab Book is the front end. We also provide engineering support for the first few experiments to help people get started. In year 2, we have ambitious plans. Among them is to provide 7 more workflows, including histology and all the trial-based analysis, and with a lot of focus on behavior. And one of the much of the focus is on providing the training infrastructure and resources. This is something that we really need to focus on. We've been very busy running projects, and exporting data into Neurodata and uploading to Dandy automatically, so that once, if you use DataJoint, the task of packaging the data for sharing is not an issue. So this is just these are just examples from year 1. So this is an example of the array electrophysiology pipeline element, and this is calcium imaging. So each step represents a table in the database, code in Python, and data stored. So for example, segmentation and calcifluorescence, it's a step in the process of either analysis for red tables, or green tables is where manual information is requested through graphical user interfaces. And we have
You've got a couple minutes left.
Okay, 1 minute. And so we have a strong process for ensuring testing, validation, and we follow this process in selecting projects. And finally, we provide services to help labs develop plans, to define these workflows for their specific needs, to help them set up either fully managed by our company or on premises, the entire process, and finally, set up processes for collaboration and publishing. This is our team, and we thank the scientific steering group in particular, but thank you very much. This is DataJoint in its current state.